Good morning, everyone. In today's lecture, we are going to discuss about the cell division process. It is a process wherein the cell divides into to form its own daughter cells. Here, the parental cell divides into two daughter cells. Cell division process occurs in two phases, that is mitosis and meiosis. In case of mitosis is also called as equational division, whereas meiosis is called as a reductional division. Here, the chromosomes number get reduced to half of its number, whereas in case of mitosis, the chromosome number remains same. Okay, so that let us see what are the different stages of the mitosis and meiosis. So before actual starting of the mitosis process, there is one more phase called as a interphase, which is called as resting phase. In the interphase, uh, the cells prepares all the enzymes, proteins needed for the cell division to occur. Okay, this it is subdivided into different stages: a gap 1 phase, in S phase, and a gap 2 phase. In case of gap 1, all the, as I mentioned, all the proteins, enzymes necessary to start this to start the replication process or any other metabolic process are synthesized in the uh, gap 1 phase, whereas S phase is also called as a synthetic phase. Here the DNA replication actually starts. So next is the last stage is the gap 2 phase. Uh, so this here the cell is ready to start the next. Here the cell is ready to start the mitosis process, and it synthesizes all the remaining substances which is actually needed for the cell division process. Okay, let us see what are the different stages of the mitosis. The first phase of the mitosis is called as a prophase. In case of prophase, the chromosomes are very condensed and compact. There is no presence of the nuclear membrane. Okay, and in case of metaphase, the chromosomes lie at the center, at particularly at the equator region. In the case of anaphase, ana means opposite. So all the sister chromatids along with its centriole, they moves to the opposite poles of the cell. That phase is called as anaphase, and the last stage or the last phase of the mitosis is the telophase, wherein you can see the division of the cytoplasm as well as the division of the nucleus into two, and there is a formation of two daughter cells. Here all the nuclear here nuclear membrane reappears, and all the cell organelles also reappears. So that's about the mitosis. In case of meiosis, as I mentioned, it is a reductional division, but here the prophase, the first stage of the first phase of the meiosis one is bit longer to when compared to mitosis prophase one. So meiosis prophase one consists of different stages: leptotene, zygotene, Pachtene, Diplotene, and Diakinesis. And friends, these are very important, and you have to remember all the key points which I am not going to discuss. So let us see the first stage, that is Leptotene. Here the chromosomes appears in the form of thread-like structures. So the chromosomes are very condensed. In case of Zygotene, there is a pairing of the chromosomes uh, through a process called as synapses, and this pairing of this synapses is brought about by the complex called as a synaptonymal complex. In case of Pachtene, we can see the crossing over phenomenon, and this crossing over phenomenon occurs between the non-sister chromatids of the homologous chromosomes. Crossing over simply means crossing of the, as you can see here, crossing of the sister chromatids of one chromosome with that of the another chromosome, particularly non-sister chromatids. Okay, next is the Diplotene stage. Here we can see the formation of the chiasma region. So after the occurrence of the crossing over, the like the region of the crossing over is called as a chiasma. The center point is called as the chiasma. You can see this is the chiasma. Okay, And in the last stage of the prophase-1 of the meiosis is diakinesis, and here we can see the presence of the bivalents in case of the cell. And still the nuclear membrane is still in the fragmented stage, and that's it friend. This is the end of the prophase-1, and let us see what happens in the next phases: metaphase, anaphase and telophase 1. It is these are the three phases of the meiosis. The same thing happens here also. Chromosomes are appear in the chromosomes lie at the center, and here there will be, in case of there will be separation of chromosomes in case of anaphase-1, and also in case of telophase-1 there is a reappearance of the nuclear membrane and nucleolus, and there is a formation of two daughter cells. You can see the division of the cytoplasm as well as the division of the nucleoplasm. Okay, next the cell, after this, after the formation of two daughter cells, the cells go into the to next stage is called, that next stage of the meiosis, that is meiosis 2. It is almost equivalent to the mitosis 1. It is very similar to the mitosis. So here you can see the prophase 2, metaphase 2, anaphase 2 and the telophase 2. The main difference between meiosis 1 of anaphase 1 and meiosis 1 of anaphase 2 is that in case of anaphase 1 you can see the separation of the homologous chromosomes, whereas in case of the anaphase 2 it is the separation of the sister chromatids. So this you have to keep in mind. It will be asked in the competitive examination, so please note down this point. So that's it friends. In today's lecture I have explained about the cell division process. So I hope you have understood whatever the key points I have tried to cover in this lecture. So if you have any doubts, please write in the comment box section, and I'll meet you in the next lecture. Till that, take care. Bye bye.